Chris?
Is it ready to start?
Yeah, it is. There you go. You're fine.
I'm actually.
There you go.
Desktop 2. Do you have any video or?
No, Chris?
Yep.
Son too.
Test.
You will hear an echo because his audio is on, so that when he plugs in, it's good to go.
Got it.
So like when you speak on here,
You're
pretty
pretty
quiet. But it's,
Sounds good. I'll ignore it. All right. Thank you, everybody, for being patient. Technology is such a time saver sometimes. Okay. Welcome on behalf of the Neurology Grand Rounds and the Department of Neurology. I want to welcome everybody to today's Adult Neurology Grand Rounds. We have a speaker invited from the University of Cincinnati, Alberto Espay. He really doesn't need any introduction. I think most of you know him from either having been here or from social media, from your interactions with movement disorder, etc. I do want to tell you that with a little help from ChatGPT, we generated a cartoon that we couldn't use for advertising because of the branding, but we can use it here. And this is what it came up with, which is not too bad, I think. But ChatGPT doesn't always give you fantastic images. It also gave us this one, which looked like we were both, being poked in the rear.
Okay.
So, not okay. But without the help of any Photoshop, I do want to share this courtesy of Bob Pascuzzi. This is Alberto when he was coaching the girls team when he was here. So he hails back to IU. So welcome. All right. So what we're going to do is we're going to test your own knowledge and our ability to move the needle on what you think about this topic before and after the debate. So if you want to participate, get your phone out, get the QR code, and you're going to be asked a couple of questions. Are proteinopathies cause or consequence of neurodegenerative diseases? And I'll give you a few minutes to punch in your answer. You can only get one answer. No multiple choice here. And show you, could be either. Oh my gosh, this is an enlightened audience. Okay, we'll measure it again at the end. So 61%, 25 cause, 14 consequence, and 2% neither. All right. So someone take a picture of that so we can compare it later. All right. So before we start the clock on the 20 minutes each, I want to propose the following for this debate. I want to ask, can we use our differences to design the killer experiment, as my postdoc mentor used to say, to answer the question or test the hypothesis? What would be the experiment that would convince you about whether it's loss of monomeric species or toxic oligomers, fibrils that cause neurodegeneration? What model would you use to test the hypothesis? Would it be animals? Would it be people? And lastly, and more importantly, it should be the first of many more discussions for this generation of scientists to have so that future generations can work on something else. So with that in mind, this is the format that we're going to follow. 20-minute affirmative case. I did give Alberto the opportunity to go first since he was a guest, and he said, no, you go first. So you get what you get. Then 20 negative case, and followed by a 5-minute rebuttal from each side, and then there'll be audience Q&A. So someone start the timer and let it ring hard so that I can hear it. So this is what we're going to be talking about. What are the key drivers of neurodegeneration in neuronal seeding models? Loss of alpha-synuclein function as an example, or amyloid, or whatever your favorite aggregation-prone protein, or is it gain of toxic function, or could it be both? And so we know that, as an example, synuclein has a lot of normal functions shown here, that many of which we don't really even understand, right? And as it goes from a monomer and it aggregates into various different structures, oligomers, fibrils, aggregates, or Lewy bodies, is it the loss of that endogenous protein that's causing the problem? Or is it the gain of toxic function as it aggregates and pulls in a lot of things alongside with it? So that's the premise, right? And what I hope to show you is that the process of Lewy body pathology formation itself, the kinetics of it, are the major driver of neuronal dysfunction and neuron loss. So that initially you have a gain of toxic function followed by a loss of normal function. And I hope to show you with some data that that correlation holds true. So neurodegeneration and protein aggregation, the things we agree on, I think, is that neurodegenerations are devastating diseases. They occur in advanced age, but also in younger people, like ALS, FTD, multiple sclerosis, Down syndrome, and that certain proteins are prone to aggregate under certain conditions. And when proteins aggregate, they cannot perform their normal function. And that protein aggregation alone does not account for the processes involved in neurodegeneration. As you all know, my bias is neuroinflammation as an example. And finally, I think we agree that we as scientists are just trying to do the best experiment we can so that we can answer the question. Things that we disagree on, and Alberto can correct me if that's not true, is that protein aggregation involves monomers, oligomers, fibrils, and sometimes inclusion formation. He will have you think that it's monomers to fibrils, okay? So I think it's more complex than that. When proteins aggregate, they can become toxic under certain, not all conditions, even when plenty of monomer remains in the cell. The process of aggregation is what matters, the kinetics and the cell types. And fibrils are not entirely insoluble, and they're not inert. They can poke holes in membranes and sequester lipids. So when he tells you that you're going to go from monomer, which is protective, to fibril, which is inert and not toxic, I hope to show you that fibrils are in fact toxic and they're not entirely insoluble. I want to acknowledge a whole bunch of people that provided inspiration and data for what I'm going to show you today, including Chrome AI, and a lot of colleagues here, a lot of colleagues elsewhere, some of whom are online, and the Tansey lab that always keeps me on my toes, questioning the underlying assumptions. So here's Alberto's biophysical model that he published recently in Bioessays. It's a beautiful model, but the problem is that it's mostly based on recombinant proteins in a test tube. And so when you think about cells, it's more complex. And when he finally gets to describing the clinical pathological framework versus the biophysical framework, he basically then admits that it is complex, but that most of the studies have used tau, and it's an N of 1, and you really can't generalize to everything. But then at the same time, he says, most of these polymorphs or aggregated proteins are very similar in the core. The surface might be different, but they're similar in the core. And then finally, what's really important is that, no, there are lots of examples, 37 human proteins that are disease-specific proteins that have organ-specific distribution, and their abundant non-inclusion alpha-synuclein pathology, for example, in Lewy body negative LRRK2 mutant cases described recently by Paul Henning Jensen. All of these have different implications for diagnosis, treatment, and future research. So let's dig deeper into this part about the lack of disease-specific protein. He says, it's unclear why subtle differences in fibril shape would lead to dramatic differences in disease phenotypes. Without statistics, standardized methodology, or mechanistic insight, clinical correlations or classifications or classifications based on amyloid polymorphs are unwarranted. So I'm here to tell you that starting with transthyretin, there are a lot of others that have been described that are very different from each other. They have organ-specific deposition. They have a lot of different effects. And understanding the specific amyloid protein and its deposition pattern is essential for diagnosis, treatment, and research. So while he says that monomers into fibrils, they're all basically the same because the core is the same, the surface might be different, he will also tell you that Alzheimer's and Parkinson's are very heterogeneous diseases, which is exactly why we need to specifically treat each different protein differently. So fibrils are not inert. They are toxic. As they form, they recruit organelles and lipids and disrupt their function. Increased cell death at late stages is associated with Lewy body formation. And I will grant you that significant accumulation of synuclein aggregates occurs before reduction in synuclein levels that are evident. So this could be gain of toxic function, but the major reduction in alpha-synuclein levels does correlate in late stages with neuronal dysfunction and cell death. So that would be loss of function. So it's really probably both. This is the model. It's super complex when you're in a cell. It's not monomers to fibrils. It's a lot of intermediate species that do very different things, and we have a lot of unanswered questions. Here are just some examples of iPSC-treated, PFF-treated neurons, and you can see clearly that the LB formation proceeds through these multiple stages, beginning with monomer to fibril, followed by incorporation of proteins, lipids, and organelles. So they do pull in organelles, and this correlates with a reduction in neurite branches, with a reduction in synapse number, with a reduction in mitochondrial membrane potential, and a reduction in autophagosomes. Okay. Additionally, we know that there's increased cell death in PFF-treated neurons at late stages associated with Lewy body formation. So they're not inert. Finally, stop the clock.
Oh, I can hear you already.
Okay. Such a time labor. Okay, here we go. Yeah. So here's the model. Alberto's hypothesis is that alpha-synucleins, loss of function, is what causes the neurodegeneration. So if this is, now it's good? Dancing.
Yep, now you gotta select your PowerPoint or
All right. Okay.
click anywhere on the screen.
Click anywhere on the screen. Soluble synuclein replacement. Action would be the synuclein will restore normal function. So when you do this experiment, you actually find this schematic that I showed you before, that the cell death, okay. So will restoration lead to less cell death? So here's the experiment: alpha-synuclein lentivirus transduction, and you would expect that you should have more soluble levels and less death, okay? What actually happens when they do this experiment is that there's actually more death with addition of soluble synuclein as the seeding process is occurring in a human hippocampal primary neuronal culture. Now, this basically means that addition of soluble synuclein, you're just adding gas to the fire because things aren't getting better, they're actually getting worse. And then the real opposite experiment is what happens if you knock down that soluble synuclein as the seeding process is going, right? You should really test both sides. So when Halal's lab does this, you actually see that knockdown of monomeric synuclein is actually improving things. So I submit to you that this model has been tested and it has not yielded data to support the idea that addition of soluble monomer is going to be a good thing. And I will stop there. Excellent. More time for discussion, yeah. Let me just lift it, yeah. Then you have to do this, then you have to do stop sharing and then sharing. I think they want me to sit and watch you. Apparently they couldn't hear me when we came back on. So make sure they can hear. Okay. It's all good. Make sure they can hear him. No, that's his. There we go.
Well, thank you for the one. And wonderful for this, and Lori for coordinating, and Bob for inspiring, and for everyone among the audience here. But contributed to a piece of who I have become. This is where things began. Not quite in this building. This building did not quite exist when I was here last. It's been 24 years since I was at AU, and it's such a joy and tremendous pleasure to be with you. I've seen this place in many different incarnations, and I love the current one, and I think that you're doing terrific. So thank you for coming and for inviting me. The concept of proteinopathy is an important one because it is one that requires for us to constantly assess the extent to which it is an idea worth holding on to. An epiphenomenon, reactive, or even protective. I'm going to take that sign up. Okay, right here. If I can, no, I cannot. Somebody can press the OK sign there. My disclosures are here. They're important because the last one is one in which it required for these ideas to be made into a company, file a patent and make a company, so that we would have the funds to do the preclinical studies to get them going. We would not be funded otherwise, because adding fuel to the fire, as the analogy was presented, is crazy. We have framed degeneration as a game, things going on. The data from which shows that there has been a loss. You can see that what X in the case of Alzheimer's, where we have both ends of the onset of symptoms, is the loss. The time at which proteins are high, as they can be measured in the spinal fluid, is decades before the onset of symptoms, both in sporadic and in genetic cohorts. This guy was one of my favorite people that I learned from, Bob Burke. Unfortunately, he died too young, but he did synuclein research at a time we were not committed to anything. We were not married to any ideas. And he actually was just the first to look into synuclein. And he found that synuclein was everywhere in the body, and it must be important in the brain. And when there was a model of apoptosis, cells destined to survive must express synuclein. And only those that would die would not. Trouble is that we then have Brack and colleagues. This is the most cited paper in the field of Parkinson's. I'm mostly going to deal with the field of Alzheimer's because we have both ends of protein homeostasis. Here we have only one end, the pathology end. But this is what's interesting. What Brack did is that he put together the brains of 41 people who had Parkinson's and 69 of people who had the Parkinson's brain, not Parkinson's history. And by the way, this is what happens in every brain bank on the planet. Most pathology-filled brains come from people with no neurological history of any kind. What Brack and colleagues did is that they put this brain in terms of their staining for synuclein from least to most and organized them in such a way that they saw a pattern. The pathology in their eyes moved from one region to another. It's kind of like if you see an elephant in a cloud, you see the elephant moving. It's pareidolia. Pareidolia is making a pattern out of static data, in this case. If Brack and colleagues were correct, then you would have expected with the data they used to create a pathological staging, the older we are, the higher our Brack staging. Similarly, if the Brack staging had any relevance to the clinical staging, the higher the pathological staging, the higher the clinical staging. That's a theory. The actual data from Brack et al. Showed anything but. And the idea is beautiful. We all fell in love with the Brack staging from the outset. Me, I fell in love head over heels. But nature doesn't give a damn if our ideas are really good and lovely and compelling. This is one of the most elegant ideas, and it is as elegant as it is false. Now, how do we define these diseases? We define them by the accumulation of a pathology. And that's why they call proteinopathies. But proteinopathy as a term means disease of the proteins. And if these proteins are everywhere in the body, they must lead schizophrenic lives because they are good for us. They must be, otherwise they wouldn't have been preserved since prehistoric genomes until they turn evil. And what happens is that most of the research, including the one that was summarized in Malu, talk about one end of protein homeostasis. So if I say proteins begin as monomeric, soluble, functional, and transition by virtue of a reaction to something into fibrillar, insoluble, non-functional, that will be most likely. At a population level, this is an inverse correlation. But if I don't tell you about one side of the story, and I mention this going up, the story that we're going to talk about is going to be one-sided. And when we have only one side of the story, it's really hard to come up with a true idea. We need both sides of protein homeostasis. Where proteins begin, where proteins end. Now, it's interesting that the field that got us into the transition cognitively of pathology as biology is the field of Alzheimer's. When we have evidence of Alzheimer's pathology, we have begun the biological journey to Alzheimer's. In fact, it doesn't matter we have no symptoms. As of a couple of years ago, if you have brain amyloid in your brain, you have biologically defined Alzheimer's. Trouble with that idea is that in a population-based study, all of us are accruing amyloid plaques as we grow, because all of us are reacting against something, and proteins are there to react for us. If you look at that data, you'll see that in fact, for most of the first 2 decades we have data, these curves are divergent. You could argue just from this data that we are able to age in the manner we do because, not despite, we accrue pathology. It is not until the ripe age of 85 years that those curves become parallel, let alone try to converge. You would think that if amyloid would have earned the adjective toxic, these curves should approach each other toward the end of a lifespan. But instead, most of us with amyloid pathology will never develop dementia. 4/5 of us with amyloid will be perfectly normal. And when the studies of odds ratio have been done in centenarians, the odds ratio of pathology in centenarians is 0.3. That is when we know that the superagers have managed to generate pathology and sustain high levels of the normal protein. So we have to keep in mind that with every story that we are reading in the literature, are we seeing both ends of the story? We have to ask that question because 98% of the literature shows the pathology, but it doesn't show us the monomeric fraction of the protein in whatever function it is. And of course, if we have that side, it's proteinopathy, we think it's the gain of toxic function. But what is missing of it all is the fact that proteins just don't evolve in any animal species so that then we would have our own built-in source of toxicity. They have evolved to be our first line of response to different agents. And that is missing from the literature because we want to still insist that Parkinson's is one disease, Alzheimer's is one disease, heterogeneous as it is. And so we kind of figure that the definition should continue to be based on the proteins, which are the consequence. Now, as I've shown you, pathology does not predict disease. Carl Harrop, a neuroscientist from the University of Pittsburgh, wrote a book that I think is an important read. He's been embedded in the field of Alzheimer's. He wrote a book called How Not to Study a Disease. And he argued that the relationship between pathology and Alzheimer's is not unlike the relationship between gray hair and Alzheimer's. Yeah, one precedes the other. So if you look at both ends, it is fair to ask, is it possible that dementia occurs when you have pathology and low levels of the normal protein from which pathology comes versus normal cognition being preserved if you have pathology plus a healthy level of monomeric proteins? And that's exactly what's been identified. You can see this is amyloid positive individuals. These are all people that have amyloid in the brains. And the question is, can the levels of A beta 42, the monomeric fraction of amyloid, predict who is going to be normal and who is going to be demented? And you can see that the lower the levels, the clearly strong the correlation is with dementia. And that happens across any tertile of amyloidosis. It doesn't matter if you have a lot of amyloid. If you continue to have high levels of a threshold of 800 picograms per ml, most individuals at any level of brain amyloidosis will remain normal. Another way to look at that data is to show this, simply to show you that that relationship is almost linear throughout, in that the levels of amyloidosis are not that well a predictive source of information. And keep that figure here because I'm going to come back to it. And the question, of course, is, well, you just show us cross-sectional data. How about prospective data in genetic cohorts? Remember, the reason we also think that the proteins are toxic is because in the genetic cohorts, they overexpress protein. So that's why we know this is the case. Turns out the exact same results happen. And this is now over a 3-year period. So longitudinal study using amyloid positive gene carriers. The higher the levels at baseline, the lower the probability to evolve into dementia, the longer the time to phenoconvert. So amyloid is common in all of us as we age, but low cerebrospinal fluids of A beta 42 is the rule. You can have many of you with amyloid plaques in your brain, you can just fine, but not a single patient with Alzheimer's would have normal levels of A beta 42. A single one. So the relationship with the monomeric fraction of the protein is tight. It's a one-to-one relationship. The relationship with amyloid, not so good. And what studies have done to knock down the protein, A beta 42, is also consequence. And what's interesting is that the mechanisms by which those consequences are, and we're going to touch on this, are the mechanisms of genetic forms of Alzheimer's. Turns out, we had a discussion yesterday about this. We had a pre-debate at dinner with Malu. And Malu might not have been aware of the fact that there is one model, actually 2 models of mice Alzheimer's, whereby A beta 42 analogs have been added on and rescue has been achieved from a behavioral and degenerative pathways. The same thing in the field of Parkinson's with a synuclein model in age rats. This is what you see here. The co-expression of alpha-synuclein, in fact, brought the function to be restored. So we have evidence that if we do this, we can do it. The investigators that were in the latter part of the study haven't been able to get further funding done because this is a crazy idea. Now, let's just end with this concept of the story of Alzheimer's from the perspective of the causal genes. We know that we have genes that are affecting these pathways. Here you have an amyloid precursor protein that is about 600 or so amino acids. It gets catalyzed by beta secretase into 99 amino acids and then beta and gamma secretase. And turns out that gamma secretase is the last step in the catalysis of this big amino acid, and that is what we think is toxic. Well, the APP mutations increase beta secretase, and then the presenilin mutations are going to increase gamma secretase activity, or so we think. So what we're going to show you is that this is an idea that's interesting. It's a gain of function toxic idea. And essentially, it boils down to the fact that the enzyme substrate complex. This is the presenilin is the enzyme that's catalytically active within this amino acid, gets in the 99 amino acid protein, cleaves it, and forms A beta 42 under normal circumstances. When there is a mutation, that enzymatic function is impaired. That's called stalling. And the A beta 42 production goes down. So in presenilin 1 mutations, contrary to what we have said to ourselves, there's a loss of function of gamma secretase and a reduction of A beta 42. And that's been shown in study after study. Most mutations of these 140 or so mutations, most of them decrease the production of the protein. It's not overproduction. And if we looked at the relationship with even other early forms of Alzheimer's, you see that the levels in mutation carriers when they develop Alzheimer's is actually lower than it is not only in sporadic, but in early onset sporadic without the mutation. There was a case report just recently presented in Nature Medicine. Nature Medicine never presents case reports, but this was an important one because a presenilin 2 carrier in his 70s was normal, despite the prediction that he should have had the disease begin in his 40s. What was about him? And these are the explanations. All the tau pathology is confined to the occipital region. Explanation for why this individual is right. Okay, so there was a paper in recently, and it's quite interesting because remember, presenilin 1 mutation, the most common genetic source of Alzheimer's, lowers gamma secretase activity. In this particular study was trying to identify what that lowered gamma secretase activity would do versus wild type presenilin 1 carriers. And it's fascinating because all of this was here in the paper. So faster increase in amyloid, okay. More rapid decreases in hippocampal volume, lower MMSE, higher CDR-SB. That is all to say worse cognitive function and very low A beta 42, which you already would have predicted. And if you look at the data from tertiles, from the lowest to the highest tertile, you see that the lowest tertile of gamma secretase activity has the steepest decline in A beta 42. And by the way, earlier age of onset. The conclusions of that paper were that we need to continue our strategy of gamma secretase modulation. Gamma secretase is a target. You would think with all this data is to increase gamma secretase function because you've seen that it's depleted and that the levels from it are going to be low. But it's not what we're seeing here. Let's do more secretase modulator trials. So where did this idea come from? Is that it? Uh-oh. Uh-oh. Do I get any credits from the interruptions? Did I get any credit? Okay. Okay. Do I have any leniency? 1 minute. Okay. So look at this. What secretase modulators do is that they decrease A beta 42, and this is what they do. But you would think that then that's going to only make people worse, according to the prediction that I showed you earlier, and that's exactly what they do. The lower levels make people worse. So it really is not the case that they increase, but they decrease the function, lower levels, and it is telling that 25 years since we have donepezil approved, we now have another drug approved, and you would think that is amazing, but instead, it has made donepezil amazing. Such that we actually cannot calculate the number needed to treat for lecanemab because there is no responder rate reported that you need for the number needed to treat, the number of people you need to treat for one to tell you he or she is better. We cannot calculate that from lecanemab. That number was 10 to 12 for donepezil. And by the way, historically, placebo has won 5 times over active interventions. We should be giving people placebo. But what makes lecanemab exceptional? In supplementary material, figure 5, increases in A beta 42. And in fact, it is the feature of most monoclonal antibodies. They increase A-beta 42. And while we know that there is across trials a relationship between how much amyloid you remove and the improvements in cognition, the same relationship it can be seen with the A-beta 42. So I'm going to leave you with these ideas here to simply say, our patients don't care who is right or who is wrong, who won the debate and who lost the debate. Our patients care that their physicians are not married to any idea and are searching for the truth. And I hope that you will never take anything at face value and that you will question everything. Thank you.
Got it. Okay. All good? Am I sharing?
Start your presentation before you share.
Yeah.
So that you can see which window you need.
Yep. All good. All right.
Click on.
Now click
Oh, yeah,
on.
it's good. It's good.
It's good. All right. So you can start the clock. I am very glad that Alberto brought up dominantly inherited Alzheimer's. And this is the human experiment to show that it is sufficient to have overproduction of proteins that are prone to aggregate, like A-beta 42 and 40. And Alzheimer's is something that develops in Down syndrome individuals because they have an extra copy of APP because of the trisomy 21. Lucky for us, it was great that the Down syndrome consortium, of which Liz Head is part of, and she's online, is now getting approval to run donanemab in people with Down syndrome because they should not be discriminated against for access to these drugs. Problem with the interpretation that Alberto has is low A-beta 42 in the CSF means it's not coming out of the brain parenchyma. It doesn't mean that it's a deficient biological condition that is causing the degeneration. High A-beta 42 in the CSF, that means it's getting flushed out of the system, beta 42, because it flushes it out of the brain parenchyma. And that's exactly what you want. Gamma secretase inhibitors, they had problems because of the on-target effects on other proteins like NOX. So most people not sure. Okay.
Application for the appointment.
No, sure. Yeah. Trying to listen to this grand rounds, ask some Alberto Espay.
All the other intermediate species that have been documented in human brains, that have been documented in animal models. Everything is homogeneous and you shouldn't have these differences in species, but you do. Differences in species, post-translational modifications in proteins, Halal, Shul, and others have shown, can make them very reactive to membranes. And in addition, you have to recognize that different brain regions have different susceptibility to these toxic proteins. Maybe there's a region in the brain that can deal with it better than others. Maybe there's more microglia in certain parts. And so you have to make sure that you know that all polymorphs, as he calls them, are not the same. They may share a core species, but the surface matters, and you actually have to acknowledge whether the resilience may be coming from the ability to neutralize some of these changes. And again, saying that the pathology has the same distribution everywhere is really not matching reality. So I'll leave you with the same conclusion. The monomer to fibril biophysical model is too simplistic. The kinetics of aggregation matter, and that you have an initial gain of toxic function followed by a loss of normal function. The fibrils are not inert. And the take-home message is that we already know that under certain conditions, protein aggregation can be toxic, but not always. And finally, there's a lot of things that we don't know and some that need to be tested in the clinic. Oh, we'll just go to the next one, which is
Okay, perfect. All right. So I'm not going to put slides just to avoid the issues. they're back and forth. So turns out, just to go on to the first point, I put the slide there for Bob Pascus to see that he can testify that in Down syndrome, there is a loss of the beta 42 over time. Patients have high levels when they're not demented. Down syndrome patients with dementia over age, levels are very low, very similar to what happened to mutation carriers. They have high levels when they're compensating, low levels when they develop the dementia. The proteins that we're talking about are really ubiquitous. The fact that they were truly preserved since we came to Earth speaks across species of the animal kingdom of their importance. And in the case of synuclein, for instance, it's actually even in red blood cells. It's everywhere. That's why you do a biopsy anywhere you can find them. Synuclein is actually made redundant with beta and gamma synuclein. These are very important proteins. Somebody said, Chris actually said yesterday that maybe I'm going to come across as a protein hater. I'm a protein lover. It just so happened that when the proteins transition into something that they aren't normally in, when they go from a natively folded to a misfolded, which is a cross beta configuration, it's a very stable configuration for the proteins to be toxic, which is why, despite what you've heard in animal models, in human studies, there is no relationship of how much and where the pathology is, how much degeneration or inflammation there is in those regions. Just none. You would think by now, with as vast of an experience with clinical pathologic studies, we would have a very clear clinical pathological correlation, and we don't. And as I mentioned, most people with pathology do not develop the diseases we predict they should. And so we've created an artificial line of research called resilience. How could it be that they have pathology and they can live with it? How toxic it is. And the word toxic is used in a knee-jerk fashion to attribute to proteins. And toxic oligomers, that idea is the mother of all ideas. And it was born out of a 2006 Nature paper that many, many years later, and over 3,000 citations later, was eventually found to be false, and it was retracted. The paper was retracted. The ideas, they are very live and well, still dictating everything. It doesn't matter that the paper was rejected. It doesn't matter that the findings of that paper that gave birth to the idea of toxic oligomers was not proven and never replicated. We're going to insist in doing that same work. All I want is to do the response to that first question that Malu posed. What would it take? What kind of study would it take? And I argue that in humans, not in animal models, if in humans we are able to increase the levels of the monomeric proteins above the compensation threshold that I mentioned, and patients do not get better, that's a false hypothesis, and let's move on. But we in neurology have the lowest rate of hypothesis falsification in medicine. We're really good at coming up with ideas, really bad at rejecting them. Thank you.
Okay, there are some questions coming in and some comments that I think are pretty interesting and probably relevant. Some of these things could obviously be adaptive, right? And we can't throw back the clock of evolution. Alberto, you can stay up here. I think it's important to point out that, you know, papers get retracted all the time, as they should. It doesn't mean that there aren't another set of papers that have supported or refuted a hypothesis, right? Science is self-correcting. So if the first or second or third or 10th paper on something is retracted, it does not and should not impugn the entire field. Let's go to the voting before everybody leaves. Did we move the needle on making you think about alternatives or maybe weighing the data in a different way? So those of you online can also vote, I believe, if they can see the QR code. I'm not seeing it on the Zoom, but we'll move forward. It could be either. So we didn't, let's see, cause was at 25, consequence was at 14. So we gained on the could be either. And hopefully that means that the kinetic model resonated, because I think that every stage of every process is going to be dynamic. And importantly, if we're going to target early, we should target what's going on. And if we target late, we should focus on what's going on. All right, 70%. Well, thank you. We can stick around a little bit, I think, for questions. The ones that came in the Zoom. In measuring CSF levels, how does this compare to postmortem tissue? Could impaired clearance impact downstream CSF levels? So the active removal of species was measured by Holtzman and colleagues, and it actually shows that you only have to get behind 30% in removal to end up with amyloid plaque accumulation. These proteins, as Alberto said, have been evolutionarily conserved, but evolution doesn't care about aging. You just have to pass on your genes. And so once that happens, the rest of it is downhill, unfortunately. Aggregation could be part of the evolved function of these proteins. Why assume they're not functional? I think that's a really good point. I think we do know that proteins become and form complexes and could potentially work as multimers. Could aggregation be an adaptive response to something else going awry if failing to restore homeostasis becomes chronic and maladaptive, like inflammation? I love that. I think that's a possibility. In chronic traumatic encephalopathy, the disease model suggests accumulation of tau. From a loss of function perspective, how would this work? You want to address that one? What happens in CTE with tau?
Yeah, so the proteins really undergo a phase transition. That was the idea of that paper that you referenced, is to think of proteins as following the laws of physics, really. It's a phase transition. So in chronic traumatic encephalopathy, tau is being phase transitioned into the pathological tau. The natural model of chronic traumatic encephalopathy is, does anybody know the natural model of chronic traumatic encephalopathy? Who said Woodpecker? Woodpecker. What does the woodpecker's brain, how does it differ from a weight-matched bird brain?
Pretty good shape.
Tons of tau. So tau is reactive, you would think, or even protective in that setting. It's kind of like a built-in helmet.
Yeah. So with regards to tau, the tau ASO clinical trials recently demonstrated that if you target the tau oligomers, you can get rid of the neurofibrillary tangles and reverse degeneration, which tells me that if you reduce the amount of species that are being recruited into toxic oligomers, you can let the neuron heal itself. And that's the interpretation that Tim Miller gave at ADPD when they presented the tau ASO. So that is also not supportive of, more tau monomers would be helpful.
Just a comment to that, though. This is why we were talking last night about what is going to take us to move ideas out and bring new ideas in, falsifying hypotheses. We have 4 anti-tau trials. We accrue 52 anti-amyloid trials. And because we're testing safety and efficacy of molecules, if a trial fails, it's not the hypothesis that we're testing the safety and efficacy of. And so at some point, we need to say, okay, we have 10 trials that are all not moving in that direction, regardless of what the animal model data says. If in humans you get to a point where you get target engagement and the results aren't what you predict, don't assume it's the molecule or the dosage or the responsiveness to the scale or the patient population being too advanced. I always joke, Imagine if our oncology colleagues would have said, well, that trial failed because the drug wasn't the right one and the patients were too advanced. We need to do the same trial earlier, and surely we'll succeed. I think we need to make sure that our trials have a hypothesis testing component. And in theory they do, but in practice they don't state, we conducted this trial to test the hypothesis. And because you don't make that a statement, the conclusions of the invariably negative list of trials is that the trials weren't good enough. We need better ideas too. We need new ideas. We need to falsify ideas that are logical, but not true.
All right. I think we will leave it at that since we're 10 past the hour. Thank you to all of you for your presence and insight. And thank you to our Zoom audience. All right.